Hello everyone, my name is Nayomi Camilus and I'm a second year master's student in the Environmental and Life Sciences program at Trent University, supervised by Dr. Sunela Mardik. Today's talk is titled How to Detect Biologicals Such as Amino Acids. So first of all, to start off, what are amino acids? Amino acids are basically molecules referring to any molecule containing an basic amino group, a carboxylic group, and a side chain that varies between amino acids. So it's a specific organic side chain that is unique to each amino acid. So these amino acids are critical to life because they're the building blocks to the synthesis of proteins for both structural and metabolic processes. My research will focus on tyrosine and its analogs, phosphotyrosine and nitrotyrosine. So these are the chemical structures of these amino acids. So tyrosine, phosphotyrosine, and then finally nitrotyrosine. As you can see pictured in these orange boxes are the different specific organic side chains that is specific for each of the amino acids. And here we see the basic structure of an amino acid containing a basic amino group, a carboxylic group, and then the R, which is the specific organic side chain that is unique to each amino acid. So now moving forward, we're going to be looking at why tyrosine and its analogs are important. As you can see here, the tyrosine is correlated with many metabolic and mental disorders. These disorders are a result of an imbalance of tyrosine in the body. As you can see, high levels of tyrosine lead to tyrosinemia disease, and low levels lead to depression, albinism, and alkaptonuria. So these are some examples of some journal article the titles of some articles. As you can see here, one of them is saying the effects of tyrosine depletion in normal healthy volunteers, implications for unipolar depression. And then you can see how all of this are very related to and showing the importance of studying tyrosine and its analogs. So now currently there are various methods to detect tyrosine. However, some of them include chromatography, high-performance liquid chromatography, electrochemical methods, spectrophotometry, upconversion, luminescence methods. Although these methods are available to detect amino acids, these methods are first have some downfall as they are tedious, they involve expensive instruments, and are time-consuming. And this here shows us that there is a need to detect these amino acids, especially tyrosine and its analogs, and the methods listed here are not really helpful or advantageous in terms of their detection. So an alternative method is required, and nanomaterials serve as the alternative method. So first you may be wondering, what is this new term, nanomaterials? I never heard of this term, but just let me break it down for you. So nanomaterials are basically particulate dispersions or solid particles with a size, the range of 10 to 1,000 nanometers. So you can compare them to very, very small particles. And these nanomaterials can actually be modified to target specific molecules. And what is another unique thing about these nanomaterials is that they have a wide range of applications, and they've been used in sensors, catalysis, environmental monitoring, and are valuable sensing materials. So as you can see, the schematic depicted here is showing an illustration of how nanomaterials are used in cancer diagnosis. So you can see how the outer part is actually modified to detect a certain protein or DNA in order to help with cancer diagnosis. So as you can see, these nanomaterials have very useful potential applications in sensing. So now my project actually focuses on one type of nanomaterials called carbon dots. So this is a very small carbon dot, so they actually measure below 10 nanometers in size. They're actually very easy to synthesize, they exhibit low toxicity, they're water soluble, and they allow for color-based testing. So these characteristics actually are very useful and advantageous in terms of using them to detect amino acids. So now my research actually focuses on to see whether carbon dots, whether they can be used to detect biologicals such as tyrosine and its analogs, specifically phosphotyrosine and nitrotyrosine. My research objectives are the following. So firstly, to test the photophysical properties of carbon dots. So photophysical property just refers to the effect of light, so how it responds in terms of light. So we want to see how these carbon dots, actually show the characteristics in terms of to the light. Secondly, we would like to study the interaction between carbon dots and tyrosine analogs, so phosphotyrosine and nitrotyrosine. And lastly, we would like to study the interaction between carbon dots and nitrotyrosine in a biological setting. So not just in a little buffer system, but we actually want to see it in terms of a biological application to see whether they can be used in a potential in the future for a potential application. So now these objectives were carried out using spectroscopy, again, which uses light to measure response. So both ultraviolet spectroscopy and fluorescence spectroscopy were used. So over here is just a simple schematic to describe how we use spectroscopy to see the response. So how this works is that these carbon dots actually have this green solution. They're green, and you can see how when we add either tyrosine, phosphotyrosine, we expect to see a response. So either a high response where nothing happens to the carbon dots, or we see a low response, as in like the actual amino acids actually decrease the light or the decrease and then show a low response. So that's what we will be measuring, and we will be showing that in the results section. So now a little bit more about the carbon dots, just the makeup of the carbon dots. These carbon dots were actually synthesized by Nakashi lab from Concordia University. They're made from glutathione and formamide. So here is the formamide and glutathione. So these were actually shipped to us, and we are very grateful to have these carbon dots for our research purposes. Now, just a little bit more about the carbon dots. So this is a vial here of these nice, very green solution of these carbon dots in water. And you can see here we tested this carbon dots using UV vis. So that's what's shown here. So these dual fluorescent carbon dots were tested using the UV vis, and figure 1a and the figure 1b shows the fluorescence spectroscopy. As you can see in the UV vis, we have some nice little bumps here, which corresponds to the different transition states. So you can just refer to them as the different bonds that are seen in the chemical makeup of these dots. And we see how they have a very nice characteristic that is very unique to the carbon dots themselves. Now looking into the fluorescence, which is more important in terms of understanding the interaction of further, if we were to study the amino acid, we can see here that there's two different distinct peaks. So there's one at around 460, I would say, and then one around 679. And here you can see that the blue one is the 460, and it corresponds to the core of the particle, while the red one, or the 679 peak, actually corresponds to the surface, or to the surface emissive states or outer part of the carbon dot. So these are very interesting characteristics of the carbon dots that we can use to study when we add the amino acids, so either tyrosine, phosphotyrosine, or nitrotyrosine, we can see how this little this fluorescence emission can actually shift depending on what is added, and that is how we will study to see the interaction between them. So now you can see here for objective 2, just studying the interaction between carbon dots and tyrosine analogs, phosphotyrosine and nitrotyrosine. You can see here that carbon dots actually show a decrease in fluorescence response when nitrotyrosine is added. So this is very, very specific. So you can see here in the first plot here, we see here tyrosine, secondly phosphotyrosine, and lastly nitrotyrosine. But we notice here that only nitrotyrosine shows a decrease, while tyrosine and phosphotyrosine really don't show much of a decrease in response. And as you can see, as a conclusion, we can take in that only nitrotyrosine is responsive to these carbon dots, which means that these carbon dots actually change. You can say that they lose their fluorescence capability in terms of coming lower in terms, and they have a low response. So they're not green anymore. They kind of lose that green solution to show a low response. So now, so you can tell that nitrotyrosine is actually the carbon dot's actually detecting nitrotyrosine, and it's very helpful. So now, just to analyze the nitrotyrosine even more, we can do a titration. So that's what we see in the next slide here. So here we can see here the different, I was telling you about the curve. So here the curve is actually shifting. As you can see, when you increase it from 0 to 0.105 millimolar, you are actually decreasing the response of the carbon dots. So the carbon dots actually go down as you increase the nitrotyrosine concentration to about 0.105 millimolar. So when you calculate this decrease, it's actually about a 22% decrease in response, and that is quite a lot in terms of actually detecting. And as you can see here, I have referred to as quenching, and this quenching basically is the same thing as referring to a 20% decrease in response. So when you take this little peak here and you plot it in terms of a nitrotyrosine concentration, we can see that it's actually quite linear, and we see an R-squared value which is very close to 1. So we can see here how when nitrotyrosine concentration increases, we can predict that even the fluorescence quenching or the fluorescence response would actually decrease. So you can imagine when we use a further higher concentration of nitrotyrosine, we expect this to further go down and have a lower response in terms of the fluorescence ability of the carbon dots. So this is very promising in terms of actually using carbon dots to detect nitrotyrosine. So now in terms of the last objective, so objective 3, studying the interaction between the carbon dots and nitrotyrosine in a biological environment, we see here there's a decrease in fluorescence observed after addition of 0.105 millimolar nitrotyrosine in both buffer and saliva. So this experiment is showing us that even if you use the nitrotyrosine carbon dots and you put it into artificial saliva, 10 times diluted artificial saliva, we still see the same response, that there is still a 22% decrease in response observed, both in buffer and saliva. And this is very useful, and because we see here that it doesn't matter where these carbon dots and nitrotyrosine are interacting, even if it's a buffer, even if it's saliva, it's still going to give you the same response in terms of the same amount of detection. There's still that same amount of detection you can do to find that nitrotyrosine is actually in the solution, and I'll be able to detect it because I have a 22% decrease in response. And this is very promising, as this is very biologically relevant, that we are not only able to see this response in the buffer, but we can also see it in a biological environment like saliva. So now to conclude, so these carbon dots are very selective and are biologically relevant. As you can see that these amino acids have different makeups, but only nitrotyrosine is able to actually interact with the carbon dots and show a decrease in response. And this decrease in response not only stopped at the buffer, but also was continued in the saliva. The same response was seen in both buffer and saliva, making it very useful for biological applications. So now, so in terms of the application component of this, as mentioned before, that these amino acids are actually correlated to many metabolic and mental disorders. So and the current detection methods are very tedious, time-consuming, and involve expensive instruments. So CDs actually could serve as a detection tool to detect amino acids, to specifically target these metabolic and mental disorders in order to have a better way of detection, and also using a better method that is less time-consuming, because as you can tell, it's very fast in terms of, these carbon dots are easy to synthesize, and they can use as potential tools for amino acid detection in the future. In terms of future work in the study is that we hope to conduct pH studies to mimic different biological environments. This will help us to see the interaction of pH and how it affects the carbon dots, the makeup of the carbon dots, how it affects the interactions between the amino acids, and also possibly to see how they actually decrease in response. What kind of interactions could they be happening between the nitrotyrosine and carbon dots that allow it to actually decrease the response, but and to understand it even further. So in closing, I would like to make some acknowledgements. I would like to thank Nakashi Lab for providing us with the carbon dots, Mardik Lab for their continuous support, Dr. Sanela Mardik for her amazing supervision and guidance in this project, and lastly, I would like to thank NSERC for funding this project. And then thank you all for listening to my presentation, and I hope you learned a lot about carbon dots and the importance of detecting biologicals such as amino acids. Thank you.